Okay, great. Thanks, Denise. So the whole concept for today's session was just for beekeepers and whoever's interested to submit some questions, and I will make my best attempt at answering them. So I mean, I've been working with bees since about 1997, on and off, but always in a research setting. So my experience is mainly from the research side, even though there's a lot of beekeeping that's involved in being a good researcher. But I mean, as a researcher, your goals are somewhat different. I've tried to answer the questions as best I can. Some of the questions I can't answer, and some of the questions I'm not even going to attempt to answer. But we'll go through here, and we'll see what we can do. And if you do have any questions during today's session, just write those down on a scrap of paper next to you, and you will be able to enter those in the chat pod at the end, and I will work on answering those as well. Suck me dry of all my knowledge, which shouldn't be too hard. So I get people from all over the country here, which is great. So this first question that I got was, I really, really like this question because it gets at the heart of a lot of the questions beekeepers have. And it's more of a philosophical question. And it's, have you noticed how in learning about beekeeping, both for a novice or someone who with a great deal of experience in trying to increase their knowledge, there is always conflicting information given by knowledgeable beekeepers. So this person grew up in an active beekeeping family, has been beekeeping on her own for almost 20 years. While the body of beekeeping knowledge has increased tremendously, the clarity has not. Pick your topic: hives, hive location, feeding, medications, equipment, best bee breed, requeening, hive manipulations, pest prevention. There never seems to be a definitive answer for anything. I always tell a new beekeeper, there is one thing you can count on: no two beekeepers do anything the same way. Why is this so? This is really a fascinating question, especially from a research side, because if you're doing research on bees, it's really not that much clearer than if you're a beekeeper. There's all sorts of conflicting information, and as a researcher, that's really my job, is to plow through these conflicting reports and try to get at what's reality, what is actually happening, and to provide some clarity. Even though there's definitely more research, I agree that the clarity is not necessarily there. And I guess this is a good preface for the whole talk in saying that there's a lot of variability out there in what beekeepers will recommend, and what has worked well for one person may not work well for another. So what I've tried to do is as much as I can, try to draw from the scientific literature to give you some answers. But back to this philosophical question about why there are so many answers. And if you have 12 beekeepers in a room and you ask a question, you're likely to get 13 or 14 different answers. And I think the real crux of the issue is that there is incredible, amazing variability between hives. And you all know this, and I have certainly seen it. Some hives do great, some hives do very poorly. And it's never exactly clear why one thrives while another one kind of manages to just hang in there or even die out. Certainly there are environmental differences between hives. One hive may have a little bit more sun, or another may be in the shade, or the rain issues, or climate, or season. There's all sorts of environmental issues that can cause variability between hives. Then, of course, there's the genetic factors. The genetic factors that a beekeeper can control, buying your queens or rearing your queens of a certain genetic stock. But again, it's hard to know exactly what your queens what kind of drones they've mated with, because almost all production queens are open mated, and it's hard to know who the father of your workers are. So there's a lot of genetic variability that's out there. In fact, bees need genetic variability to thrive. And then there's diseases and parasites. One colony may have more diseases or parasites than another, that increases the variability as well. And who knows what other kind of variability there is out there. Effects of, I don't know, phase of the moon, who knows? There's the potential for incredible variability between hives, and I think it's clear to all of us that it exists. And it's a major challenge when you're trying to draw any sort of conclusions, either through research or through beekeeping, about what's good and what's bad. So here's just a case study, and this is something since I've done some research with pesticides and looking for pesticide effects on bees. This is a good case study to look at the variability and the reason you get so many different answers. So if you treat bees with a pesticide and you want to see how many bees are affected, one of the simplest ways to do that is with a simple dead bee trap. And we've got dead bee trap strapped to the front of this little cardboard nuke here. This is just a two-frame nuke that I was using, just a bare minimum colony. But you can capture the dead bees using a device like this dead bee trap here. Now, I had 20 to 30 of these little cardboard nukes set up, and before I even treated them with anything, there was already incredible variability between these two-frame colonies. I mean, how much variability is there possible in two frames? Well, we saw it. You get, and even between days, there's incredible variability in the number of dead bees you trap in these little colonies. This is actually a very important thing. How many bees do pesticides kill is a major topic of research, because it's important to know if pesticides are actually having a negative effect on bees in field situations. So there have been research done on the number of bees that die naturally in each colony on a daily basis, and we've got 3 to 3.5% of your foragers will die every day. That's just normal. So if a pesticide is increasing the mortality of bees, a small effect would be like a 7% mortality. Essentially, you're doubling the normal colony mortality. A medium effect would be like a 17.5% daily mortality. And then a large effect would be 30 to 35%. And this is really getting up into the catastrophic range of forager die-off, because if you're losing 35% of your foragers every day, that colony will die. That's just all there is to it. So 35% is really catastrophic losses for a honeybee colony. So the question is, how many colonies do you have to include in an experiment if you want to be able to reliably detect this 35% loss of foragers? So this is catastrophic level effects that you're looking for. This is not some sort of subtle effect. We're looking for the worst possible effect. How many colonies do you need? The answer is, so they did this modeling experiment to see how many hives you would need to reliably detect a 35% loss, daily loss of foragers. And you would need 74 hives to reliably detect catastrophic losses using just the dead bee trap. And this is really humbling to me as a researcher. It's because these honeybee colonies are so variable, you need a huge number of hives in order to be able to draw conclusions. 74 colonies to detect what should be pretty obvious, and I'm sure if you looked at the colonies through some other method would be obvious that these colonies are dying. But if you're just looking at dead bee traps, you would need 74 colonies to be able to reliably detect this really incredible mortality from poisoning. And now if the effect is more subtle, if you're just losing 10% of the foragers, well, then you need 500 hives. And if the effect is even more subtle and it's just barely above control mortality, well, you're well into the thousands. So the reason you need these huge numbers is because honeybee colonies are so variable. So the take-home story message from this is it's both difficult and expensive to do good experiments with honeybee colonies. To get the kind of clarity that beekeepers are looking for, you need to run experiments with huge numbers of colonies. And I mean, this is really outside the realm of possibility for most beekeepers, and it's difficult even for bee researchers. These are expensive experiments. And this is the reason there's so little clarity, because it's so hard to do experiments that will provide the clarity that we all need. So I guess what we need are better mechanisms of looking at honeybee health and survival and improved methods beyond just that dead bee trap that I showed you to hopefully reduce some of that variability and collect more data from fewer colonies, or we need to set up these huge experiments with lots of colonies. And I would encourage any beekeepers out there, if you want to start drawing conclusions, you need to do experiments on a lot of colonies. Because all 13 opinions can't be right. Most likely there is one answer that is correct. It's just that we haven't done the experiment to demonstrate what it is that's correct yet. So there were a lot of questions that we received, going on to the questions now. There were a lot of questions that we received about queens, and the success of queens has become, seems to be a pretty big issue in beekeeping these days. So here's a representative question. When I was expecting hives this spring, I found 15 to 20% queenless hives with no sign of hive swarming. Do you have any idea why queenlessness was such a problem this year? Recent years, queenlessness has really, I think, climbed up the ranks among the problems that beekeepers are having, and unfortunately, I don't really have a good answer for this. I know that the scientific studies that have been done, David Tarpey at North Carolina State University has been interested in this question. And they published a paper in the Journal of Economic Entomology just this year. And down at the bottom here, you can actually get a copy of his paper. And what he looked at was the health of commercially raised honeybee queens. Most of these were raised by the big queen breeders out in California. And they got these queens from the California breeders, and they measured everything they could about them. They put them in their own colonies, and they let them raise brood. They measured the queens. They assessed how many sperm were stored inside the spermatheca of these queens, which is a measure of the mating success. They looked at the viability of those sperm. They assessed the number of different drones that the queens had mated with. And a lot of this is because much of the suspicion is that queens are having trouble these days because they haven't been properly mated out in the almond orchards of California, where these queen breeders are doing their work. But what they found is that's not the case. The commercially reared queens had plenty of sperm. They had, as you can see in the abstract here, they had, almost 5 million stored sperm in their spermatheca with a viability of 80%, which is fine. These queens had mated with an average of 17 different drones, which is better than the 12 drones that David Tarpey previously reported in an earlier study. So it doesn't seem that it's mating that's the problem with these queens. And from their measurements, it's not the size of the queens. They all look to be full-sized, well-mated queens. There's no physical reason that they should be failing as much as people have been reporting. So it doesn't seem to be anything in the queen rearing process or the queen mating process that these bee breeders are doing. They seem to be perfectly good queens, at least in everything you can measure this way. So it's not really clear what the problem is. It could be that something in the breeding program that these queen breeders are using is introducing something that causes increased queen failure, or it could be some sort of sublethal exposure to something that's killing off the queens, or it could be some sort of low-level disease or some sort of pathogen issue that could be causing them to die prematurely. It's just really not clear what the problem is with the queens in this country and why we're having so many problems. I wish I had a better answer for this, but David Tarpey is really the expert in this field, and he can't seem to find anything that's wrong with the queens that people are getting from the California breeders anyway. Now, they did notice that there are, even among, though generally these queens are good, there is a range And some queens are better than others. Some queens are bigger. Some queens have more sperm than others. So there are queens that are less good, but they still should be adequate. And the vast majority of the queens were fine. Certainly nothing to give us the impression that there's some obvious problem there. So there's a big question mark for you. More research needs to be done in this field, is I guess all I can say. Here's a question that's probably better for Denise, but we talked a little bit about this, and I'll try to answer it. Please list the number of perennial flowering plants favored by honeybees in particular. So just to give you some pretty pictures, I asked Denise what her favorite bee plant would be, and she said this blue vervain, which has this spiral flower, and you can see the bees just love it. I believe this is in the mint family, and Denise may disagree with me on that. I don't remember. But it's a beautiful native plant. Bees love it, and I believe it's a perennial, so it'd be a good if you're going to plant things for bees. This is a good thing for you to plant. My personal favorite is the pussy willow, and I don't know why I like pussy willows. I know they're excellent for bees because they're one of the first flowers that blooms in the spring, and they're really an excellent pollen source. So this is the first new pollen that a beehive can collect early in the spring is likely to come from pussy willow. So if you can plant pussy willow around your apiary, this will give bees a nice pollen source early in the year, right when they're really in need of pollen to rear all that brood that is going to collect. That's really going to get your colony started in the spring. So pussy willow is a good thing to plant as well. And I just kind of, I don't know, I've always liked pussy willow. So that's my personal favorite. I also like dandelions, but that's not really a perennial or anything that you would be planting intentionally, but another important plant for honeybees. Of course, there's lots and lots of different plants out there, and we've got really a national audience, so it's hard to say what's best for one particular place. But if you go to the Bee Lab website, and this is beelab.osu.edu, and this is just a screenshot of the front page, and if you scroll down below what you see here, there will be a link to a sheet Denise has put together with links to the different resources you can get for plants to plant for pollinators. And there's all sorts of people have lists, and hopefully you can find something that's appropriate for your goals and your area of the country. There's quite a bit of interest in this topic, so some resources for you. If you have a smartphone, there's also an app you can download. This is for the Android phones. There's this Bee Smart Pollinator Gardener app that you can get from the Google Play Store if you have an Android smartphone. And you just put in your zip code, and it will tell you what kind of plants you can plant for your particular area. And it's really a neat little tool. And I believe it's also available for iPhones, so you can go take advantage of that. This is put out by the Pollinator Partnership, and it's really pretty well done. So moving on for plants, questions about swarms and absconding. So we got a couple of these. So I'd like tips for encouraging caught swarms not to abscond, or not to abandon their new hive. I caught 3 swarms this year, and all 3 abandoned my hives. One I put in a brand new top bar hive, identical and located beside the hive it swarmed from. It left after 1 week. The other 2 I put in Langstroth hives with some used, but I believe clean comb already built. I fed all 3 hives, and they appeared to be eating fine, but they left a day later. I don't know where they went. Any tips for encouraging them not to leave? Well, this is a tricky question. I caught a swarm this spring, and I also had it abscond, so you're not alone. It happens to everybody. So, I mean, number one, of course, is be sure you've got the queen. If you don't have the queen, well, the swarm isn't going to stay, and I imagine most beekeepers look to try and make sure they have the queen in there, or the bulk of the bees into that box as they shake them in or clip off the branch and put them in their equipment. Also, you want to be sure to use some old equipment that smells like bees and will help, give those bees someplace to start. They've got queen someplace to start laying eggs in there. You don't want to put them all on foundation or just on empty top bars. That's a hard place for bees to begin to make a home. They kind of want you to do some of the work for them already before they move in. So give them some comb that's already drawn out. Give them some honey or some food to help them out. I know if they're too hot, I'm trying to think, because a swarm is really the same thing as a package. Fundamentally, a swarm and a package are the same biological condition for bees. So we don't usually have that many problems. Generally you don't have problems with packages leaving after you install them. So I don't know if it's that swarms are generally caught later in the year and they might be getting a little hot. You want to make sure that they're not stressed environmentally. They're not out in the blazing sun where they might have a problem. And I don't know, you could consider confining them in some way. I haven't tried this, and I don't know if this is a good method or not, but you could, in some experiments we've been doing, it's amazing how you can confine bees and they will do just fine without being able to free fly as long as they have food in there. You could confine them for a period, just like we do with packages, and that would definitely prevent them from leaving. They could overheat in there and die from other causes, but then you would have your dead bees confined as well. So it's an issue, and it's something I'd really like to learn more about. Is how to keep how bees choose their home is an interesting topic, and Tom Seeley just published that book about how honeybee democracy, about how honeybees decide on a place to live, and us, we're trying to convince them to move into what we're providing, and I guess sometimes it's not always what they're looking for. Why that is, I don't know. So the next question: are Japanese hornets a threat to Ohio's beekeepers? And this is a very interesting question to get to the bottom of. The short answer is no, but it's an interesting story. So here is the Japanese hornet, Vespa mandarinia, and this is a substantial hornet. These can be up to 2 inches long or bigger. They are supposed to have an extremely potent sting that is quite painful. And there's a subspecies, Vespa mandarinia japonica, that is native to Japan. And it causes a lot of problems for the beekeepers there, actually, because this particular hornet, well, like all hornets, it is a carnivore, and it eats other insects. And it collects other insects to feed to its own larvae. And the Japanese hornet likes to feed on honeybees and use honeybees and honeybee larvae in particular to feed to its own larvae. And just a relatively small number of these substantial hornets can get into a honeybee hive, and they can kill off all of the bees in the hive in a relatively short amount of time so that they can be free to take the honeybee brood and feed it to their own brood. It's a pretty grisly way for a bee colony to die. Yeah, 55 millimeters long. Japanese hornets, however, exist only in Asia, where they are a real problem for beekeepers. And they kill the bees to feed to their own larvae. And here's an interesting website down at the bottom if you're interested in these hornets. They have a good deal of information on this German website about hornets. But these hornets are only in Asia. So we don't need to worry about them here, at least not at this point in time. And there's always the chance they could get introduced at some point. But they're not a problem today. Now the Japanese, it's an interesting story in coevolution in that the Japanese subspecies of the Apis cerana, the Asian honeybee, is actually adapted to dealing with these Japanese hornets. So in the center of this ball here, this ball of bees, is one of these Japanese hornets. And the Apis cerana japonica's natural defense for dealing with these hornets is to ball them and to produce heat by shivering their wing muscles. And they basically overheat the hornet that's in the middle of this ball. Well, it's not just heat. They also use CO2 because all these bees are respiring and producing CO2 And not only do they increase the temperature, they also increase the concentration of CO2 in there, and up to a level that the bees can just tolerate, but is too much for the hornet, and the hornet will die. And this is their natural defense against these hornets in the Apis cerana japonica subspecies. Now, Apis mellifera, the European honeybee, doesn't have this long history with the Japanese hornet, and it does not do this. So it really is defenseless against the Japanese hornet. So apparently, if you want to raise Apis mellifera in Japan, you need to use some sort of hornet trap in August in order to keep these hornets out of your colonies. So this is definitely not something we want to introduce to the United States. They're large and pack a potent sting, so there's all sorts of reasons not to want them. I guess they could maybe control some sort of pest insects, but I don't think it would be worth the downside to introduce them. Now, there is another, not technically a hornet, it's the Asian predatory wasp, and this is a wasp. It's also a good-sized wasp, 30 millimeters in size, and it also feeds on honeybees as well as other insects. It's a carnivore. Now, this Asian predatory wasp was introduced to France in the year 2004 from China, probably in a shipment of dishes that was shipped to France. And it has spread out from France. It's now present in northern Spain. And it can be a problem for the beekeepers over there because it, like the Japanese hornet, will feed on bees. And it's such a big wasp that it can just kind of get in the beehive and do whatever it wants because it's so huge. And again, the European honeybees have no defense against this kind of thing. But again, Asian predatory wasp has not been introduced to the United States, so this is not a problem here. There are some hornets and wasps in the United States. There is the European hornet, which was obviously introduced from Europe years and years ago, Vespa crabro, and it, on occasion, can invade honeybee colonies and start to eat honeybees and larvae because that's what hornets do, is they eat other insects. But I don't know that it's a particularly large problem. There's also the bald-faced hornet that I've seen in bee colonies on occasion, and I've heard that yellow jackets will sometimes enter honeybee colonies to rob out as well. The other wasp that you're likely to find, and I know we get calls about it fairly frequently, is the cicada killer wasp. And this is, again, this is a substantial wasp. 50 millimeters, it's a big wasp. But this is really not an issue for honeybees. These cicada killers are solitary wasps. They're ground nesters. And again, they eat other insects, cicadas especially. And they really have no interest in honeybees. And they can't recruit their sisters to really invade a colony like some of the other wasps and hornets can. So this is an impressive-looking insect, but it's really not an issue for honeybees, and it's not an issue for people either, generally, because they're not aggressive, and you can just they might try to get in your face, but you can just ignore them because they're generally going to leave you alone. So so those are some hornets and wasps that are problems elsewhere, but not in the United States. Here's another issue, Tropilaelaps. What is Tropilaelaps, and should beekeepers in the United States be worried about it? So Tropilaelaps, I don't know if anyone's ever heard of this. It is another mite, also from Asia. In fact, it shares a lot of similarities with our old nemesis, the varroa mite, which you see on the left here. Varroa mites are kind of saucer-shaped, or sometimes I think kind of UFO-shaped. They're kind of little alien things. But they're much larger than the Tropilaelaps adult mite. It's kind of more elongated and about a third the size of varroa. Now, currently, we do not have... Oh, so here's a picture of a Tropilaelaps mite on a giant honeybee larva. So this is Apis dorsata, another species of bee that lives in Asia. And the Tropilaelaps is native on this giant honeybee. And with the introduction of Apis mellifera, this mite has also moved over to the European honeybee, where it can cause some serious problems, same as Varroa. So Tropilaelaps is similar to Varroa, but smaller. It reproduces just like Varroa. It lays eggs under capped brood cells, but its life cycle is quite a bit shorter. I mean, it's a smaller mite, so it can reproduce more frequently. Its life cycle is shorter, so it can build up to large numbers quicker. So it can really devastate a colony over a shorter period of time. So this is not something we want to see in the United States. I mean, I guess the good news about Tropilaelaps is that it can solve your Varroa problems because it will kill your colony first. It can outcompete Varroa. So rather than having colonies die of Varroa, you could have them die of Tropilaelaps because the Varroa were outcompeted. It's not much, not really a good outcome, but you can detect these the same ways you would detect Varroa, with a sugar roll, ether roll, or sticky board. You're just looking for these mites that are about a third the size of the Varroa mite. A big difference between Varroa and Tropilaelaps is that the Tropilaelaps mite cannot feed on adult bees. It requires brood to survive. So a Tropilaelaps can ride around on an adult bee for a couple of days, but it can't actually pierce the cuticle of the adult bee to feed on the adult bee's blood. So it will starve to death in a few days if there is no brood in the colony. So if colonies have a broodless period every year, that would be a big help in controlling Tropilaelaps. So if Tropilaelaps is introduced to the United States, it's predicted that it would be more of a problem in southern climates where brood rearing takes place year-round. In the north, where you have winter, it's predicted to be less of a problem. But given its quick build-up time, it's hard to say. And really, one of the control techniques that you could use for Tropilaelaps is to confine the queen for a period and cause a broodless period during which all of the Tropilaelaps mites will die because they can't survive. Formic acid and apistan are effective controls for Tropilaelaps. And the good news is Tropilaelaps has not yet been found inside the United States. There is a large survey being done by USDA APHIS to assess the status of Tropilaelaps in the United States, and some of you listening may have been participants in that survey. They have not found it here. So we are still Tropilaelaps free, and hopefully we will stay that way for many years to come. So Tropilaelaps is not a concern at this point, but we do still need to worry about our old nemesis, Varroa. So another question is which miticides and mite control strategies are effective? There are a number of strategies and products out there on the market. The chemical controls include apistan, checkmite. This is not on the market anymore, but some may have some in their shed somewhere. Hivestan. There's amitraz, which is not used legally in the United States anymore. Thymol, Apolite var, and Apiguard. The organic acids, oxalic acid and formic acid. And this new one made of hops, HopGuard, made from hops beta acids. So which ones are effective? We've got all these things out there. Which ones are actually effective at increasing survival of honeybee colonies? Well, again, this is I'm going to take some survey results from the Bee Informed Partnership, and you can go to this website and look at these results yourself. This is the same group that is doing the Tropilaelaps survey, so you can look at their results on that. And they just basically surveyed a lot of beekeepers across the United States and tried to discover how they're managing their bees and what success they're having with those bees' survival. And the results they're presenting are survival from fall 2010 to spring 2011 here. And those are the results I'm going to show you. So formic acid is effective at increasing colony survival. So on the y-axis here, we've got average colony loss in percent, and unfortunately, average colony loss is right around 30% over 2010, 2011. But in the colonies that used where formic acid was used, there was a significant decrease in colony loss compared to other miticide products or nothing being used. So formic acid appears to reduce colony losses on a yearly basis. And I would interpret that as it doesn't have any, the benefit from the mite control outweighs any sort of negative effects that the product may have on brood, because you're getting increased colony survival. And that's really what we all care about, is that colonies can actually make it. So formic acid appears to be effective. Apiguard, which contains the active ingredient thymol as well as some other essential oils. Same story. There is a decreased colony loss with Apiguard use relative to other products or nothing. Apolife Var, again, another thymol-based product. There is decreased colony loss on a yearly basis relative to other products or nothing. Now, in this survey, they asked about a number of other products and strategies: checkmite, apistan, sucricide, powdered sugar, mineral oil, other herbal treatments, drone brood removal, small cell size, and screen bottom boards. None of these caused a significant decrease in colony loss on a yearly basis. Now, it's not to say that some of these may not be effective in controlling varroa mites, because some of them, not a lot of people reported in the survey that they used some of these methods. So the sample size for some of them is small, and it's difficult to detect significant differences with a small sample size. But I'm just reporting here what they found to be significant and the things that I would say are now proven to work in 2010 and 2011. I should mention, of course, fluvalinate and coumaphos used to be very effective, but the mites have generally evolved resistance to these compounds. They did not survey for HopGuard, which is really a brand new compound, or oxalic acid or amitraz, because these two cannot be used legally for varroa control inside the United States, though they have been shown to be effective in research settings in other countries, though I don't know if there's any sort of survey data like the Bee Informed Partnership presents here to back that up. On to another question. So this is another chemical-related question. I would like to have your opinion on the benefits of providing essential oils with the 1-to-1 sugar that I've been feeding my bees for their winter stores. For example, is there a possible correlation in reducing the varroa mite level in my hive? Is there some benefit in reducing any harmful intestinal pathogens? And finally, would a winter store of honey made from these essential oils provide any additional benefits through their winter feeding? I do realize, however, that the honey made from nectar is the best, but these supplements, which are now appearing in various publications, appear to be a real benefit to bees. So we're talking here about the essential oil supplements that you would add to sugar water when you're feeding bees. This is different from the essential oils that are in those mite treatments, which are essentially applied to the colony in either in a pad or in some sort of gel form which evaporates inside the colonies. This is actually, you're adding it to the liquid feed. Well, there's been some work done on the effects of essential oils on bees. So this is an old paper by Detzel and Wink from 1993, and you can get the abstract here, but I've just put part of their table 1 up here. And they tested different essential oils from a variety of different species of plants. And in this column over here, they indicate whether these essential oils were a feeding stimulant or a feeding deterrent. And I was surprised to find that, you know, most essential oils are actually a feeding deterrent. So they would actually decrease bees' uptake of sugar water. The exception is this citrus oil and the caryophyllene oil, or caryophyllene, which is a terpenoid, do stimulate feeding. And that here they've got the dose they're using, the effective dose. And over here, they did find that some essential oils are toxic to bees, like caryophyllene. Even though it stimulates bees feeding, it is somewhat toxic to them. So that is not a good choice. So you're really left with this citrus essential oil, which contains a lot of limonene. And I believe limonene is one of the main constituents in these supplements that you buy, like Honeybee Healthy, because it is a proven feeding stimulant. And thankfully, it does not appear to be toxic to bees. But I would be cautious just throwing essential oils into your bee feed, because many of them are feeding deterrents, and some of them are toxic to bees. So you do want to be cautious about what you put in there. This is from the Bee Informed Partnership Survey again, the year-on-year survival of colonies using Honeybee Healthy, which is one of these supplements that's added to sugar water. And they found no significant difference between colonies where Honeybee Healthy was used and where it was not used. Now, it is a little bit lower here in colonies receiving Honeybee Healthy, but that is not a statistically significant difference. So there is no effect of Honeybee Healthy on decreasing colony loss. So there may be some benefit to the feeding stimulation with these products, which could, if you're worried about bees taking up their food, it could encourage it. But the effect long term does not seem to be substantial, or at least not significant. And I should note here that there were substantial numbers of colonies in this survey, almost 100,000 colonies that used Honeybee Healthy, and 170,000 that did not. So as I was saying at the beginning, you need at least 74 colonies to do a valid experiment. Well, we've well surpassed that number here. So I would say that this is really a definitive answer on this, and that there is no significant benefit to the Honeybee Healthy. So it seems to me your money and labor might be better spent elsewhere on your hive management than in these feeding stimulants. I mean, there's a little bit of evidence that some of these things may suppress some pathogens, but I think part of the problem is that that's generally at higher concentrations. Now, clearly, essential oils are effective in controlling varroa, but that's at a much higher concentration than you could ever get inside a sugar water situation, because you can only get so much of these essential oils into sugar water. They're just not very soluble in water, which is why when you're controlling mites with them, you have to use this vaporization thing with the gels or the pads in order to get enough in there to actually kill the mites. So I guess my sense is that it's probably not worth your trouble. Bees will take plain sugar water, okay, but sometimes you may need to add a little something to help them locate that. And in that case, these products might be useful, but I don't see any great benefit to them. Here's an interesting question. What percentage of DNA do humans and bees share? Well, the last common ancestor that us and honeybees shared was about 600 million years ago. So clearly we've had a long time for our DNA to diverge. So here is a figure taken out of the honeybee genome paper that was published back in 2006 in the journal Nature, and there's a web link for that if you're interested. Now, one of the problems with 600 million years of history is that our DNA has diverged a lot, and it's really difficult to give a percentage difference. Like, you can say that humans and chimpanzees share, what is it, 98% of our DNA. It's really difficult to do that sort of thing with honeybees and humans because there's so much time that's passed and things have gotten so different. And what this figure shows is over here we've got humans in this column and honeybees here, and you can see that this is actually looking at genes that humans and honeybees share, and these genes listed in black down here, represented in black, are genes that humans and honeybees share, and we have very clear. It's very clear that both honeybees and humans have these same genes because they are genes that code for proteins that are essential for just basic biological function. Because if you see You know, the fruit fly and mosquitoes and chickens and fish all share these same genes. So there's a lot of genes that we do share with honeybees. Looks like probably about 3,000 genes that we share with honeybees on a one-to-one basis. There is a suite of other genes where we may have more copies of them than honeybees, or there's other stories going on. As you move this thing up, there's only a relatively small number of genes that are just really honeybee or insect specific, and a relatively number of small genes that are human or vertebrate specific. It's really a lot of genes that all of us share as living things that really are the basic building blocks of life that we all need in order to be alive. All the things involved in metabolism and all the things that we do just to maintain life. We all need these genes down here in black and blue. So it's hard to say an exact percentage, but we do share a lot of genes, and that's why insects and honeybees can be good model organisms for, say, medical research. Because if you're interested in some sort of disease that is important and is affecting one of these genes down here in black, well then the honeybee or the fruit fly, it makes a great model organism for that particular disease. So that's, we share some things, but we don't share everything. Clearly, we're very different from honeybees, but there's more similarity than you might expect there. So one more question here. Have queens ever been raised from an egg absent the other bees in a laboratory setting? And the answer to this is a resounding yes. There was this research that was just done in 2011, published in Nature, showing that royal actin, this is a protein that is in royal jelly, is responsible for making larvae into queens. So if you fed them casein, which is just a protein that has no effect on bees, it's just a milk protein, you get workers, whereas if you feed them royal actin, you can actually rear queens in the same laboratory setting. Now the way you rear bees in a laboratory is in these 96, or in a plate, basically. So you have these plastic plates, and it's just like queen rearing. You graft larvae into these wells in these plates, graft a larva in here, and then you feed that larva royal jelly, actually, and it will develop into a worker. And it was always kind of puzzling why you're feeding these larvae royal jelly in these plates inside the lab, and you end up with a worker at the end of the whole process. And it turns out that this royal actin is the key, and that royal actin actually breaks down very quickly in royal jelly. So if you store royal jelly, it will actually lose the royal actin in the royal jelly, and you won't get a queen. So in this paper, they added back in the royal actin, and they got queens. So you could, if you wanted to invest a huge amount of time and effort into rearing queens or honeybees, you could do it in a lab in these plastic plates like this, and you can get both workers and queens from royal jelly. Just one last question here, and then we'll see what other people have to say. And this is a real simple one. What are your guidelines, temperature, wind, humidity, etc., for opening a hive and manipulating frames? Well, my guideline is, if it's good foraging conditions for forager bees, it's good conditions for handling the frames. And that is generally over 55 degrees Fahrenheit with wind less than 50 miles an hour. Humidity is your main concern for brood survival. If brood dry out, they will die. But as long as you can keep them humid, they should be no problem. And you can generally just set them out in the open for a few minutes, and they won't have a humidity issue. The one issue with opening them, overheating is generally not a huge problem, because bees like to be kept very warm. They actually maintain their nest at about 95 degrees Fahrenheit. So heat is generally not an issue, but you do want to keep your frames out of direct sunlight, because wax will melt at 144 degrees Fahrenheit. And I know I've taken some of these black plastic foundation frames, and if you lay those out in bright sunlight, that black plastic will soak up heat, and you will get wax melting. So that's the last question. I hope that I've answered these questions, and I'm sorry I didn't get to all the questions people submitted. But if anyone has any questions they want to submit into the chat box on the right there, I'd be happy to take a shot at them and tell you at least my answer to the question.
Reed, this is Denise. I'll just jump in for a second while folks are maybe entering some questions. That verbena that I love, that blue vervain, it's Verbena hastata. If folks are looking for it, is a perennial, a native perennial. Prefers some moist, more moist locations, but will do well in drier, but needs full sun, and it is in the verbena family. So FYI, if anybody's looking for that.
Thank you for the correction. I should have looked that up. But beautiful plant.
And it will reseed, so you can kind of share it with beekeeping friends as you have it established. It kind of spreads itself around. Susan asked about posting answers to other questions, and I do appreciate we got lots of good response to the call for questions, and our intention is to be able to answer those and post those periodically onto the Bee Lab website. In the future, I'd like to have maybe a regular newsletter that quarterly or something that we post on the Bee Lab website. So I do have intention to get to those other questions. So Susan, thanks for asking. How about Joe's question, best time to split a hive?
I mean, I guess it depends on your goals. Obviously, earlier in the season would be better so that those splits have a chance to go through the summer and build up to full-size colonies that will overwinter. That would be my recommendation, is as early as you can. I mean, in April, once your overwintered colonies are starting to build up and you're looking to prevent them from swarming, a great swarm prevention technique is splitting them. And then you'll have more colonies, build those up during the summer, and hopefully get them to survive through the winter. So next question.
Last one, let's talk about mite away quick strips, and maybe you...
So mite away quick strips are the formic. There are a few different formic acid products. You can do like a homemade formic acid formulation, which is effective but kind of dangerous because formic acid is caustic. So you can get these mite away quick strips, which are pads that you put into the colony, and you don't need to remove them. The bees will get rid of them themselves, but those do contain formic acid. So mite away quick strips should be an effective varroa control technique and would be associated with increased survival on a year-on-year basis. The one thing I should warn, though, is that there is a temperature limit. You don't want to use mite away quick strips if the temperature is going to go above 85 or 90, because they will evaporate too quickly and will actually be toxic to the bees as well as the varroa mites, and you can get some pretty nasty bee die-off. Even though it seems they recover from that generally, but do use them with caution in the summer temperatures.
Reed, how about a quick question from Ken, who asked about the zombie fly?
Oh, they found this out in California, and this is a fly that apparently gets into bees somehow and alters the bees' behavior so that the bees fly at night. And you can actually collect honeybees at a black light or at a porch light at night because it's infested with this fly that actually lives inside the bee. So the fly is manipulating the bee's behavior so that it is dispersed away from the hive, presumably so it can go infest other colonies. Now, this sounds like a horrible new thing, but the thing is, it's not new. This particular fly has been in the United States forever, basically. And they found it on wild bumblebee colonies, where it also does the same thing, presumably. I guess we just finally got lucky enough to find it. So it's out there. I guess check your porch light. If you see any bees flying at your porch light at night, you just want to grab those and put them into a jar. They'll die, but what you want to do is just wait to see if any little tiny flies emerge from that bee that you collect at night. Very interesting thing, but I don't think it's a serious threat to beekeeping. It's just more of kind of a biological curiosity, which is really interesting, but not a big threat.
Great. Well, thank you, Reed. This was a really excellent presentation. Folks, if you